Today we're gonna talk about staining. Being a microbiology student, staining is the most important thing to know. Why it is so? Okay, we are having microorganisms all around us. Okay, but why aren't we differentiating them? Why can't we see them? Because most of them are either semi-transparent or transparent, and in order to differentiate them from their surrounding, we need to create a contrast, right? As I said, most of the organisms are either transparent or semi-transparent, but there are certain microorganisms which are having natural pigments in them. For example, S. aureus, which is golden yellow in color. Another one is halobacterium, which is pink in color. But what I mean to say, if they are pigmented, they can be differentiated. But if they aren't pigmented, we are applying them color, we are giving them color. That method of giving them color with the help of chemical compounds, which are known as stains, is known as staining. That entire process is called staining. Okay, now let us list out some of the benefits of staining. First one, to differentiate, we can differentiate them from their surrounding. And second one, as they are differentiated, we can come to know that they are made up of what shape, like whether it is spherical, whether it is having pointy structures, so on. And we can even measure its size, right? Next one is understanding its chemical composition. Let us understand this with an example. If I take a plant cell has cellulose in its cell wall, okay? And taking another part of a plant cell, for example, its nuclear membrane, which will be having lipid. Okay, so when a stain, which is a chemical compound, applied to a plant cell, that stain will react differently with cellulose, as chemical reacts in themselves and give out some of the other outcome. Okay, so the outcome with the stain reacting with cellulose will result in different outcome, and stain reacting with a nuclear membrane will result in different outcome. So by studying it, by analyzing it, we can come to know its chemical composition of a cell or either a microorganisms. Okay. Next thing, we can come to know its internal as well as external structures. And we can also come to know whether they are live or dead. If they are dead, they will be reacting in different ways. If they are live, they will be reacting in their obvious manners. Okay. We can even come to know about its motility structures, appendages, so on. Here we are having 4 different main types of staining. Why it is so? Because each and every microorganism is different in its physical and chemical manner. So they have their own requirements. So to fulfill their requirements, to stain them properly, we are having different types of staining. Starting with the simple stain. Simple stain, name itself suggests that there is a simple process of staining, as it includes only one chemical compound. Simple stain includes one chemical compound for staining, so it is known as simple stain. But it do have 2 different types. First one is direct staining and second one is indirect staining. Okay, understanding direct staining, considering a bacterium which will be having negative charge. If I provide a basic dye to this bacterium, the bacterium will uptake that basic dye. Okay. So, bacterium uptaking basic dye. So, this bacterium will be colored, right? And the surrounding will be uncolored. So, it will, this type of staining helps us to differentiate this microorganism from its surrounding. Ultimate goal of staining is that, right? And so this is known as direct staining. I am bad with my left hand. And this one is demonstrating indirect staining. Here this bacterium is having negative charge, and if I provide acidic dye or stain to it, it won't uptake. Okay. So the provided stain and dye will remain in its surrounding. So here also, bacterium will be differentiated from its surrounding, as surrounding is colored and this bacterium will remain uncolored. So this is known as simple staining. These are the types of simple staining. Next one is differential staining. Differential staining helps us to identify the types of organisms, okay, from a group of organisms. The most famous type one is gram staining, which help us to differentiate from gram positive and gram negative from a bunch of microorganisms. Okay, and the second type of differential staining is acid-fast staining. This also helps us why we can firstly differentiate those gram positive and gram negative, as they are having some or the other different type of structure in their cellular mechanism or cellular constituents. So the differential staining helps us to identify that difference, so we can differentiate those microorganisms, right? So that is all about differential staining. Next one is special staining. Okay. As I said, each and every microorganism is different in their own way. Some of them have certain structures which can be stained under normal condition. Okay, best example is capsule staining. Capsule is something, if I am considering this as my microorganism, this is my microorganism and its surrounding. So this microorganism will produce certain slimy matter which is known as capsule for protecting itself. So if we want to study this microorganism, this capsule, we can't study under normal conditions, as a normal specimen, a normal slide is prepared by taking the culture, diluting it into water, distilled water. But this capsule is water soluble, so we can't dilute this certain certain culture, as capsule will be diluted into water. So it will require certain other type of method to get stained. Okay, so these types of staining fall under special type of staining category. It also has certain other types like metachromatic granule staining, then cell wall staining, nuclear staining, then endospore staining. It also has one bacterial staining in it, spirochete staining, which are generally found in tartar of your teeth. I'll be talking about it later. Okay, so this was all about special staining. Last one is vital staining. Okay, vital staining is something where microorganisms are alive till they are stained. They are basically having two different types. In one type, one is known as intravital staining and second one is known as supravital staining. Okay, in supravital staining, while the microorganism are staining, they are alive, but after staining they will die. And in intravital staining, they will be still alive even after staining. Okay, what is that different thing in vital staining and these types of staining? Okay, basically when a smear is prepared, a specimen is stained, they are primarily heat fixed in most of the cases, in not all of them. So while those heat fixing, what is heat fixing first? Why from a burner slide is passed 2 to 3 times for heat fixing those microorganisms. That is ultimately killing those microorganisms. By heat fixing, we are fixing its protein and its other molecules. Okay, that is ultimately death of microorganism. But while in vital staining, there are two different types. As I said, in one of them, after staining the microorganism will die, and in another one it will still remain alive. While in other stainings, before the stain is applied, the microorganisms are dead. So this is difference between vital and rest of them. Okay, so this was all about outline of staining. In next video, I will be talking about it in detail, about a differential, special, vital staining. Okay, so stay tuned for the next video. And if you like this video, then please do like, comment, share and subscribe to Friendtor. And if you are looking for some math content, then please do check out our channel and stay tuned for the next video. Thanks for watching.